So hello student, today I am going to discuss with the application of animal cell culture. So in this topic we generally discuss the application in which sector the application are most widely used by using of animal cell culture. So we understand, firstly we understand the definition or it may be defined as per me. The cell culture can be defined as the process of cultivation cells and tissue outside the body of an organism called in vitro in an artificial environment, which stimulate the in vivo conditions such as the temperature, nutrition and protection from microorganisms. Right. So cell culture is a process which are cultivating the cells or tissue outside the body, which outside the body of an organism in an artificial environment, which is called animal cell culture. Right. Now come to the topic, that is the application of animal cell culture. So basically application of cell culture or animal cell culture is one of the major tools to use in the cellular and molecular biology, which are providing excellent model system for studying the normal physiology and biochemistry of cells. Example, metabolic study, etc. Right. So basically, the most if I am going to talk about the application of cell culture, so cell culture is a major tools used in the cellular and molecular biology, providing excellent model system for the studying the normal physiology and biochemistry of cells. Right. In other terms, the application of cell culture, the effect of drug and toxic compound on the cells and mutagenesis and carcinogenesis. It is also used in the drug screening and development and large-scale manufacturing of biological compounds. Example, vaccines and therapeutic protein. So, in the applications in other area, right, that is the major advantages of using cell cultures for any of the applications is consistency and reproducibility of the results that can be obtained from the using a batch of colonal cells. Right. So here I am going to discuss the various sectors which are used, here I am going to discuss the application of animal cell culture into the various sectors. That is the first area is the model system. So cell culture. So first here I have written model system. So cell culture are used as a model system to study basic cell biology and biochemistry, to study the interaction between cells and disease causing agent like bacteria, viruses, to study the effect of drug, to study the processing of aging, and also it is used for the study triggering for the aging. Right. So cell culture or animal cell culture are most widely used into the model system to study the basic cells. So first is to study the basic cell biology and biochemistry. Next is to study the interaction between cells and the disease causing agent like bacteria and viruses. So it also study by using model system effect of drug, and to study the process of aging, and also it used for the study triggers for aging. So next area is the cancer research. So next area of animal cell culture into the cancer research. Right. So the basic difference between normal cells and cancer cells can be studied by using animal cell culture technique. So after applying the animal cell culture technique, so we can differentiate the normal cell and cancerous cells. So as both can be cultured in laboratory, so normal cells can be converted into the cancerous cells by using radiations, chemicals and viruses. So thus the mechanism and cause of cancer can be studied. Cell culture can also be used to determine the effective drugs for selectively destroy only cancer cells. Right. So by applying the animal cell culture, so we can use to determine the effective drug treatment for cancerous cells. Right. Now I'm going to discuss next area, that is of application of animal cell culture, that is the virology. So virology, animal cell culture are used to replicate the viruses instead of animals for production of vaccines. Right. So in virology, the animal cell culture are used to replicate the viruses instead of animals for the production of vaccines. So cell culture can also be used to detect and isolate viruses, and also to study growth and development cycle of viruses. It is also used to study the mode of infection. So in the field of virology, the animal cell culture are help into the various degree, like it help in the replicate the viruses instead of animals for the production of vaccines. It also help, the cell culture can be also used to detect the and isolate viruses, and it also study the growth and development cycle of viruses. It also used to study the mode of infection of viruses. Now come to the next area, that is the toxicity testing. Fourth, that is the toxicity testing. So animal cell culture is used to study the effect of new drugs. Right? So basically animal cells can be used to study the effect of new drugs, cosmetics and chemicals on survival and growth of number of type of cells, especially liver and kidney cells. Right? So culture media or culture animal cells are also used to determine the maximum permissible doses of new drug. So by applying the new animal cell culture, we study or we find out the permissible doses of new drugs. Right? Now come to the next area, that is the vaccine production. So vaccine production, cultured animal cells are used in the production of viruses, and these viruses are used to produce vaccines. For example, vaccines for deadly disease like polio, rabies, chickenpox, measles and hepatitis B are produced by using animal cell culture. So the animal cell culture are also used into the vaccine productions for the deadly disease like polio, rabies, chickenpox, measles, etc. Right? Now come to the next area, that is the genetically engineering proteins. Genetically engineering proteins. So the animal cell culture are used to produce commercially important genetically engineered proteins such as the monoclonal antibodies, insulin and hormones, as much more. So by using animal cell culture, we are formed genetically engineered protein which are generally necessary for various type of diseases, and we, by using the genetically engineered protein in the production of monoclonal antibodies, insulin productions, hormones and etc. Right? Now next area of application of animal tissue cell culture, animal cell culture, that is the replacement of tissue and organ. Replacement of tissue and organ. So, animal cell culture can be used to replace tissue or organ. For example, artificial skin can be produced by using these techniques to treat patients that burns and ulcers. So, however, research is going on the artificial organ culture such as liver, kidney and pancreas. Right. Second thing, that is the organ culture technique and research are being conducted on the both embryonic and adult stem cells culture. These cell cultures have capacity to differentiate into many different types of cell and organ. It is believed that learning to control the development and differentiations of these cells may be used to treat variety of medical conditions. Right. So, replacement of tissue and organ, another area for the application of animal cell culture. Now, let's come to the next area, that is the genetic counseling. Genetic counseling. So, genetic counseling in the fetal cell culture extracted from the pregnant woman can be used to examine the abnormalities of chromosomes, gene using karyotyping, and these findings can be used in early detection of fetal disorder. So the animal cell culture is also used as a genetic counseling in various fetal cell culture. Right. Now come to the next area, that is the genetic engineering. So, cultured animal cells can be used to introduce new genetic material like DNA or RNA into the cells. So these can be used to study the expression of new gene and its effect on the health of the cells. So insect cells are also used to produce commercially important protein by affecting them with the genetically altered baculoviruses. Right. So genetic engineering, the animal cell culture also used into the culturing animal cells can be used to introduce new genetic material like DNA and RNA cells, right, into the cells. Now come to the next area, that is the gene therapy. Gene therapy. So, cultured animal cells can be genetically altered and can be used in gene therapy technique. So, first cells are removed from the patient lacking of functional gene or missing a functional gene. These genes are replaced by the functional gene, and altered cells are cultured and grow in laboratory conditions. So these altered cells are introducing into the patient. Another method is by using viral vector. Functional gene is inserted into the genome of viral vector, and then they are allowed to infect the patient in the hope that the missing gene will be expressed with the help of the viral vector. So the various techniques are used for the expressing of altered gene or expression of the missing gene, so they can easily expressed with the help of viral vector, and by the help of animal cell culture are widely used into the gene therapy. Next is the drug screening and development. Next area of application of animal cell culture is drug screening and development. So, animal cell culture are also used to study the cytotoxicity of the new drug. This is also used to find out the effective and safe dosage form of new drugs. It also used to test being conducted on 384 and 1536 cell well plates. Cell-based assay plays an important role into the pharmaceutical industry. So basically these are used, the various drug screening and their development. So the role of animal cell culture are most widely used because they are help into the screening of cytotoxicity properties of new drugs. They are also used to develop an effective and safe dosage form of new drugs. Now come to the new area, that is this is the new area for use in the pharmaceutical industry. I hope you understand all related to the application of animal cell culture. Thank you for this session.